Today, I'm going to present our recent results on the use of SPRi imaging for research and clinical purposes. We are part of Gnocchi Foundation, which is an important non-profit organization especially devoted to the treatment and rehabilitation of patients affected by chronic and debilitating diseases, like, for example, multiple sclerosis, Alzheimer's disease, and Parkinson's disease. More in details, the Laboratory of Nanomedicine and Clinical Biophotonics is mainly focused on the use of innovative methods for biomarker detection. The mainly used instruments are surface plasmon resonance imaging and Raman spectroscopy. In addition, our research is oriented to nanotechnology-based detection strategies that most of the time are coupled with both Raman and SPRi imaging. For more info, please visit our website. Now, after giving you a short introduction about SPRi imaging technology, I'm going to show two examples of our recent research results obtained by using SPRi imaging. The first is the study of the interaction between LL37 and different beta amyloid forms involved in Alzheimer's disease. The second is a SPR study performed to develop and to optimize a biosensor strategy for the detection of circulating antibodies in allergic patients. Surface plasmon resonance is a label-free optical method that allows to detect the absorption of a target molecule onto a gold surface previously functionalized with ligand molecules. The approach is simple and is normally based on the fact that the physical and optical properties of a thin gold layer surface change when molecules interact on it. These changes can be detected when a source light directed into the gold surface and passing through a prism is reflected with different angles when ligand molecules interact. If we observe the reflected light at a fixed angle, we can see in real time the increasing of the light during the molecular interactions. Therefore, SPR permits the real-time monitoring of molecular interaction in a label-free approach, thus permitting ideal conditions to perform kinetic and affinity studies. A recent evolution of SPR is SPRi imaging, which can be performed when multiple ligands can be spotted on the chip surface, thus permitting the simultaneous monitoring of several interactions at the same time thanks to a CCD camera. This approach is particularly advantageous to study and to compare different ligands, considering that the interaction can be monitored at the same time and using the same experimental conditions. This is different from standard instruments, where only one or two ligands can be studied at the same time. In addition, in case of SPR-based methods for molecular detection, this approach permits the quantification of several molecules with multiplexing features. Now, I will show the study of the interaction between LL37 and different beta amyloid aggregation forms. LL37, called also cathelicidin, is an endogenous antimicrobial peptide contained in neutrophils granules and expressed in several tissues, especially in bowels and brain. This protein shows particular biochemical features, including a positive net charge and basic features, with an isoelectric point around 11. In addition, it is reported that LL37 is overexpressed in Alzheimer's disease patient brain, and that in mice, the analog of LL37 protects against the formation of amyloid fibrils found in Langerhans islet in patients affected by diabetes type 2. On the other hand, we have here amyloid beta, which is the main actor of neurodegeneration in Alzheimer's disease, due to the formation of pathogenic fibrils and plaques. Surprisingly, beta amyloid shows complementary biochemical features, including negative charge and acid properties. This and other considerations triggered some questions about a possible interaction of these two small proteins. It is possible that LL37 and amyloid beta are molecular partners. If yes, may LL37 play a role in Alzheimer's disease etiopathogenesis and progression? These electron microscope images are among the first experimental data to prove this hypothesis. One of the most clear evidences is that LL37 used at micromolar concentration is able to destroy or to inhibit the fibrillar structures made of beta amyloid. You can see on the left that LL37 alone forms a small spherical precipitate in solution. In the middle, you can see that typical amyloid beta fibrils are produced in vitro. On the right, you can see that LL37 mixed with amyloid beta in the semi-identical experimental conditions inhibits the formation of amyloid fibrils. Starting from these premises, a SPR imaging study was proposed to verify this interaction and to study whether LL37 shows some preferences for different amyloid beta aggregation forms. To doing that, LL37 have been injected onto a SPR imaging chip spotted with different beta amyloid forms. About the protocol, first of all, a bare gold SPR chip was cleaned by plasma generator. Then the chip was coated with a special copolymer developed by Marcella Chiari and colleagues. This copolymer was developed for microarray applications, but works properly also on gold surfaces. The copolymer includes three functionalities. The first and most abundant is the methylacrylamide, DMA, represented in yellow, which permits the interaction between the copolymer and the substrate surface. The silanols, MAPS in blue, were originally added for the interaction with glass surfaces, but plays here a good stabilizing role. Finally, the succinimide in red is the active moiety which permits the covalent binding with primary amines in protein. There are several advantages of using this polymer as chip coating. First, the chip coating itself can be performed just by the immersion of the chip in the solution without any other reaction. The second is that the copolymer can be modified accordingly with different experimental needs. In addition, the copolymer shows very good anti-fouling properties. Thereafter, different forms of amyloid beta are being directly spotted by using a piezoelectric spotting system. After an overnight incubation, a blocking step was performed using ethanolamine, followed by washing and drying steps. Three different beta amyloid aggregation forms have been prepared by solubilization in particular buffers optimized on purpose, and by applying different incubation time. The aggregated forms were then characterized by capillary electrophoresis. In this image, you can see the different amyloid forms and relative negative references spotted on the SPR chip. The first amyloid form was produced using the 40 amino acid beta amyloid peptide. This is the less fibrillogenic form and was here spotted immediately after its solubilization. The result is the formation of small molecular weight oligomers as proved by capillary electrophoresis. The second form was prepared using the 42 amino acid amyloid, typically related to fibrils in Alzheimer's disease. Also in this case, the peptide was spotted immediately after its solubilization. As shown by capillary electrophoresis, we have here spotted an equilibrium between low and high amyloid oligomers. Finally, the third beta amyloid form was produced using the same 42 amino acid beta amyloid sequence but incubated for 24 days before spotting in order to permit the formation of higher molecular weight aggregation forms as can be observed on the electropherogram. Here on the right, we can see the plasmonic curves of the three amyloid forms and of the reference spots measured before the SPRi experiment. What is important here is that the three different amyloid forms show different curves. In particular, the pink and the blue curves, related to amyloid 42 and containing more aggregates, look quite different if compared with amyloid 40 in green, representing a less aggregated form. After some optimization tests on the SPR chip, we injected LL37 10 micromolar in a standard buffer. Then we firstly verified the interaction between LL37 and beta amyloid forms that can be here observed. The injection of albumin and the injection of a scrambled version of the LL37 confirmed the specificity of the interaction. The next step was the calculation of binding affinities between LL37 and different amyloid forms by performing a calibration curve. As you can see from the shape of the sensogram, the interactions studied here are quite complex, and they could not be fitted with standard first-order fitting, this probably due to the complexity of the oligomeric features of amyloid spotted on the chip. Therefore, for this experiment, the association and dissociation constants could not be easily calculated. On the other hand, after verifying that all the binding curves reached the equilibrium at a certain point, and that a complete regeneration was obtained after each injection, an equilibrium analysis was successfully performed by plotting the SPRi imaging signal related to all the concentration injected and by fitting the calibration curve. Here we can see the calibration curves and some results of the SPR study. Looking at the calibration curves plotted here with different scales, we will first have a look at Rmax, which indicates the maximal theoretical response and which is somehow proportional with the amount of interacting ligands on the chip surface. The first information that can be here appreciated is that Rmax increases going from less aggregated forms to more aggregated forms. This suggests that independently by the affinity on the same surface area and injecting the same amount of LL37 molecules, we have more interaction with the more aggregated forms, probably because of the presence of more beta amyloid molecules packed together due to the formation of larger aggregates. On the other hand, here we see Kd, which is the equilibrium constant related to the overall affinity. What is not obvious is that the affinity goes to the opposite direction if compared to Rmax. As we can observe here, the affinity of LL37 is higher for small oligomeric amyloid forms and lower for bigger aggregation forms. This may suggest that LL37 inhibits fibril formation by shifting the equilibrium toward amyloid monomers and small species. In other words, this data may suggest that the higher affinity of LL37 toward smaller amyloid species may permit the sequestration of small oligomers, thus reducing the fibril formation and thus having a protective role. In conclusion, our SPR imaging study permitted to confirm that LL37 specifically interacts with amyloid beta species and that LL37 binds with more affinity small amyloid aggregation forms, thus suggesting a potential fibrils inhibiting function. To confirm the involvement of LL37 in Alzheimer's disease, the next studies will involve experiments in mice, Drosophila mosquitoes, and postmortem human brain samples. I would especially thank my colleagues at Fondazione Don Carlo Gnocchi for this study, the group from the National Research Council, the University of Pavia, and Stanford University. For more information, please read the article. In the second part of this presentation, we want to show the optimization of the detection of circulating immunoglobulins related to cow milk allergy. This is just an example of the detection of circulating antibodies that could be also applied to other clinical issues, like for example autoimmune diseases studies and drug resistance monitoring studies. Alpha-lactalbumin is one of the major allergens present in cow milk. The full alpha-lactalbumin allergen, immobilized on a microarray surface, is commonly used to diagnose and characterize milk allergy by detection of specific immunoglobulin E. Screening different alpha-lactalbumin epitopes can further characterize different groups of patients by defining the specific epitope they are active to, in order to define allergy subtypes and personalize allergy vaccines. Therefore, 8 different potential lac epitopes have been screened in a previous study, and some of them have been defined as specific allergen for different groups of patients. In this study, we have synthesized 3 of these epitopes in the form of peptides to generate a peptide microarray for IgE fingerprint. In order to take advantage from epitope-specific microarray and in order to correctly define the specific epitope recognized in each allergic patient, we need high sensitivity and high specificity. For this study, the goal was the optimization of the peptide presentation on a fluorescence-based microarray assay to be used with clinical samples. Surface plasmon resonance was therefore used to study in detail the kinetic of the interaction between different epitope presentation and specific antibodies. At this concern, the most general and common issues in the field of antibody detection are related with the correct capturing of the antibody by immobilized peptides. This is mostly related with the correct epitope presentation on the biosensor surface, both in the case of fluorescence or label-free based approaches. Even if the scheme on the left represents the ideal epitope presentation, most of the time the ligand's presentation is more likely as represented on the right. The presentation of ligands can be heterogeneous, overcrowded or poor, or a mix of them. All of these conditions produce reduced analytical performances. For these reasons, the strategies used here to improve these performances are both the epitope orientation and the multi-epitope presentation. For what concerns the epitope orientation, here we used the same copolymer mentioned before, but functionalized with azido-clickable groups. The azido groups specifically react with the alkeno groups previously conjugated with the epitopes during their synthesis. These click reactions are chemoselective, fast, and very efficient, thus permitting, first, the correct orientation of the ligands, and second, the control of the ligand's density considering that the reaction is concentration dependent. Details about the advantages of using this type of surface chemistry are reported in the publication reported here. In addition, in this study we mostly focused our attention on the multi-epitope presentation. We were wondering whether different epitope architectures may improve the antibody detection in biological fluids. In details, we wanted to verify whether the use of linear or branched or tandem architectures are related to changes in affinity and/or kinetic properties. The study of different epitopes architectures were tested by SPRi imaging using click chemistry surface activation and by spotting different epitopes architectures by piezoelectric spotting. Just to resume the experimental steps. First, we deeply cleaned the chip using a plasma generator. Second, we coated the gold chip with the copolyazide and by chip washing and drying. Then we directly spotted the different ligands on the chip by previously adding a copper reagent into the spotting buffer for the click reaction. Finally, the chip was incubated overnight, washed with EDTA in order to remove copper cation in excess and right. Using this approach, we spotted different epitopes and a combination of different concentration and architectures. For obvious reasons, we present here the result related to only one single epitope, presented with different architectures. After having verified a good chip surface regeneration, and after having verified the possibility to correctly fit the binding curves, we have been able to perform a multi-cycle kinetic study in order to obtain the affinity and the kinetic binding constants. We injected here increasing concentration of antibody anti-lactalbumin onto the chip spotted with epitope LEC1 presented by linear, branched and tandem architecture. The concentration used here are from 1 to 500 nanomolar. The regeneration steps were performed using an increasing volume of glycine and from pH 2 to pH 1.5 according to the increasing signal. Considering the high intrinsic affinity of this antibody that can be immediately appreciated looking at the binding curves, we had to use some particular experimental conditions. First, we previously optimized the ligand concentration spotted on the chip in order to avoid to inject too much quantity of analyte for the kinetic study. In this case, 100 micromolar spotted ligands permitted to have good signals without using too much injected analyte. Second, we used here a relative fast flow rate of 100 microliter per minute in order to avoid mass transport effects. Third, we had to monitor the dissociation curve of the less concentration for around 4 hours in order to be able to calculate the dissociation constant. In addition, we injected several times buffer solution in order to correct a slight baseline drift occurring during this experiment. In this slide are shown all the binding curves already corrected by the reference channels and corrected by the buffer responses. As you can see here, both binding curves and kinetic constants are quite different among the three architectures. In details, the first evidence is that the R max, proportionally related to the number of potential interactions with ligands on the surface, is much higher for the tandem configuration. This evidence, if we suppose that immobilization efficiency of the three different epitopes is equal, suggests that on the same surface area much more epitopes are exposed due to the tandem configuration. On the other hand, it is interesting to see that the branched configuration, which present two epitopes at the same time as the tandem one, does not show the same behavior, probably due to steric effects. In this slide, we can observe the behavior of dissociation constants. We can see that the antibody associates faster with the linear form if compared to the other. And this could be related to a better epitope accessibility related to the linear form. Otherwise, by observing the dissociation curves and their relative constants, we said that dissociation is very slower in case of the tandem configuration if compared to the other. This means that the interaction, once established, is much stronger if compared with other structures. The equilibrium dissociation Kd constant, which can be used to summarize the overall affinity of these interactions, confirmed that the higher affinity is derived by the use of the tandem configuration. The high overall affinity, and in particular the very slow dissociation rates of the tandem configuration, could be explained by formation of polyvalent interaction between one antibody and two epitopes. This, as already reported by Whiteside and collaborators, is normally related with slower association and much slower dissociation rates. These data have been confirmed by the fact that the detection of circulating anti-lactalbumin antibodies from serum of allergic and non-allergic patients, detected on standard fluorescence microarray bioassay, improved using multi-epitope presentation. In details, considering here the LEC1 epitope only, we can see that the significance of the separation between allergic and non-allergic subjects depend by the epitope presentation architectures. In fact, the tandem configuration show more significant result than branched and linear configuration. In this case, the linear configuration of LEC1 did not show enough significance for the recognition of allergic subjects. In conclusion, we have seen here that the epitope architecture may strongly influence both binding properties and consequently diagnostic performances. We observed here that in this study the tandem configuration is the favorite one. About experiment aspect, in case of high affinity interaction, a proper experimental design must be followed. Finally, the use of SPRi imaging has been particularly adequate for study multiple ligand and multiple spotting conditions as in this particular case. This permitted to obtain kinetic data from several epitopes by automatic sample injection on the same chip. I would especially thank my colleagues at Fondazione Dognocchi, from the National Research Council group that coordinated this study, and from San Raffaele Hospital for the subject samples and for clinical support. And thank you for your attention.